Hello everyone, my name is Mika Gustaffson. I'm an associate professor at Linköping University in Sweden. I will today talk about deep learning for identification of disease genes. So our analysis has often been to work with the protein interaction network as a backbone for integrative modeling. A key problem with this kind of approach is that the protein interaction network is biased, incomplete, and yet a hairball. For this purpose, we have worked out different analysis tool and an analysis pipeline. I have that at our GitLab page, but I would like to just pinpoint one concept that we will talk about today. This is about disease modules, which is using co-localization of the protein interaction networks when you have RNA-seq data, and that this reveals a functional representation of the disease, where you have also identified causative genes or GWAS associated genes, you can say upstream genes, which we have worked a lot with, and that we will now map to neural network field. And you doing that, a very good thing with neural network is that you can infer a representation directly from data and then analyze that representation. And for this purpose, we have used autoencoders in this work. So now I will move to talking about this publication, which we had last year together with Jesper Timmer here at KAUST on autoencoders. First, we asked ourselves, using a data set of like 50,000 samples or so, can you compress it to lesser nodes? And yes, you can. Of course, others have shown that as well. But if you use a kind of simple autoencoder, which has just one intermediate layer, you need about 1,024 hidden nodes to compress 95% of the variance. However, if you allow for some hidden layers more, so if you have 3 hidden layers, one in the decoder and one in the encoder, you can get away with just 512 hidden nodes. And it scales good also if you go below that, if you have some complexity in the model. So we work with this 512 hidden nodes and 3 hidden layers, and we asked ourselves, can we associate attributes to the different layers using light-up technique? So for each hidden node, we associated genes in the output space using light-up. And analyzing this first at the different layers, we analyzed genes in the output state that were associated with the same hidden node. So in doing that in the different layers, we found that genes that were associated to the same hidden node in the first hidden layer, they also tended to be close in the protein-protein interaction network and also be associated with the same functions in terms of biology. This was also so in the second hidden layer, but to a lesser degree, and in the third hidden layer, we didn't see this at all. So it seemed like the first hidden layer contained something that we have sometimes called functional modules in the systems medicine field. Doing the same for diseases and cell types that we had in the data, we found that they cluster mostly in the third hidden layer, and to a lesser degree in the second. And using a technique we call reverse training, we could associate diseases to output nodes. We call it reverse training, and this revealed output genes that are associated to each disease. And interestingly, these were also highly enriched for GWAS genes in the third hidden layer, but not in the first and the second hidden layer. So doing this, I think we have shown that using unbiased techniques, we can identify functional units in an autoencoding way. And we think that this is something that we should explore even more. I'd like to thank my funders a lot. Thank you for listening.